All right, y'all. So, welcome to Exam 2 Review Part 2. So, just a reminder, the exam has three big areas. That's going to be metabolism, and so that includes enzymes and then all the metabolic processes. So, the respirations and fermentation. And then we have microbial growth and then controlling microbial growth. And that's broken into the physical and chemical methods and antimicrobials. Okay. So in the first review session, we spent a lot of time on the antimicrobials, and then we started a flow chart for microbial growth. So today we're going to start with enzymes, and this is just going to be a jumping off point. My in-person classes, we did this a little bit something similar, I don't know, a week or so ago, but it's a good way to test what you remember. And so what I want you to go ahead and do is take just 1 minute and try and label the enzyme. Remember, there are two sites on an enzyme that you need to remember. So use the information provided. Trust me, you have what you need to answer this question. And identify the two sites, and then try and answer part A and part B. And there is flexibility in these answers, okay? And then I'm gonna use this as a jumping off point to talk about enzymes in general, okay? So I'm gonna mute for about 30, 45 seconds and just go and give it your best shot. Label the enzyme and answers A and B. All right, so we're gonna try and make this one a little bit more interactive than yesterday, since hopefully my sound is still working. Okay, so can somebody remind us, what are the two sites on an active site? The two little binding regions, what are they called? There we go, active site and allosteric site. Good. Which one of those sites is where the chemical reaction actually happens? Good, the active site. So if the active site is where the reaction occurs, okay, which one is going to be the active site? The triangle one or the, I'm going to say circle. I know it's not a perfect circle, but like the more round one. Good. So remember, the substrate can only bind the active site. So if I tell you your substrate is a triangle, you have to say, okay, which of these sites would a triangle fit in? And so in this case, I'm going to label it going inwards. Oh, not actually. Okay. Come on, computer, you can do it. I tested this before we started to make sure I wouldn't have any problems. Okay, hold on a second. Oh, I think I know. Oh, crud. Okay, hold on. I gotta switch my screens real quick. I have to have Word open on the monitor in order to draw on it. Sorry, y'all. Hold on. This is my husband's setup, not mine, so it's a little backwards. Okay, so let me unshare and let's try this again. Sorry, y'all. Okay, let's see if this will work for me. There we go. Okay, so because my substrate is a triangle, we know the active site must have a similar shape. So then by default, we know this other region must be the allosteric site. And remember, the allosteric site, the whole point of it is regulation. Okay, so some sort of inhibition or activation. Now, we don't talk about activation in this class. But just like we can turn enzymes off, we can also turn them on, okay? All right, so if we know the substrate binds to the active site, we identified the active site because it has a similar shape to the substrate. Think about what competitive inhibition means. Can somebody define competitive inhibition for me? Good. So competitive inhibition means there's an actual competition with respect to the enzyme. So the only place worth actually competing for is that active site, because that's where the reaction happens. And so any competitive inhibitor is going to also have to mimic the shape of the active site. So really, you could have made a million different drawings, but essentially it can be like a smaller triangle. Heck, it could even, just be something that fits in here. So it could be kind of like this for a shape. I mean, honestly, anything that would fit into that space. Okay. Normally it's going to be very similar to the substrate. I was being a little dramatic in what I drew on the enzyme, but the point is that it has to be able to kind of wedge itself in there and stay there. Because remember, with competitive inhibition, the inhibitor is competing with the substrate for the active site. Okay. And then, as Jordan mentions, the inhibitor is physically blocking that active site. So the substrate cannot get in. So if the substrate can't get in, the reaction can't happen. Okay? So then with allosteric inhibition, we know that the inhibitor has to go to the allosteric site. So this is also called non-competitive inhibition, which is how I teach it. So I'm sorry, I should have typed that instead. So non-competitive inhibition is also called, wow, I butchered that as well. It's been a long day, y'all. We ended up going all the way out to Liberty Hill to look at houses. I found the house of my dreams. It's only, like $150,000 above budget, but whatever, no problem. It's only in Liberty Hill too, so it's not even a real option. But I got home like 20 minutes before this started, so I've been kind of throwing things together. Non-competitive. So sorry, I'm a little scatterbrained. So what a non-competitive, get it together. Non-competitive inhibitor. Y'all, this word is gonna defeat me today. Non-competitive inhibitors are going to bind the allosteric site. So y'all, if you're struggling with keeping all these inhibitions separate, make little notes here for yourself, okay? So it needs to, mimic the allosteric site. All right. And so we see it's this little roundish shape. So the inhibitor could just be small enough. Let me erase my little notes here. So it could just be small enough to wedge itself in there, right? Could be a nice little round shape. Or you could have drawn something that's longer, right? That would like fill this whole region. I don't know why I feel the need to color it, but we're going with it. So basically any shape that would fit in that area. And so then remember with non-competitive inhibition, all right, when the molecule, the inhibitor binds, it's going to cause the whole enzyme to change shape. And in doing so, when that whole enzyme changes shape, what's going to end up happening is this active site is going to be warped. And so now the shape of the active site isn't going to work for that substrate. So it's not going to be able to bind anymore. Okay, so again, the purpose of all these inhibitions is to make sure the enzyme cannot do its job, the reaction cannot happen. Okay, so it does that either by competing for the active site, in which the inhibitor looks like the substrate, which by default looks like the active site, or non-competitive inhibition is where the inhibitor is going to look like the allosteric site. By binding the allosteric site, the whole enzyme changes shape and the active site gets altered in such a way that the substrate doesn't fit anymore. Do we have any questions about those two types of inhibition or inhibition in general? Okay, so real quick, just to keep the conversation going on enzymes before we move on, it's not quite as intense as the other topics. What are some features of enzymes? Because y'all, this is one of my favorite topics, so you know you're gonna see it on the exam. What are just some general features? Excellent, Garner, good. These are, for the most part, they are proteins. And that is why their 3D shape is so important. What does it call? Oh, Taylor, look at you. Y'all, look, yes, good. Okay, what happens if the protein falls apart? What do we call that? When a protein loses its 3D shape and is no longer active. Good. Denaturation is the process. We say the protein is denatured. Yeah, so good. Yeah, it's all about just verbiage and how you say it. Good. So remember, denaturing is bad, okay? Because the protein no longer has its shape. It cannot do its job, okay? So good. I saw some other things. Specificity, that's important, y'all. So an enzyme will only do one reaction. So let's say it's in charge of taking product A and converting it to B. Then that is all the enzyme does. So that means it's specific for substrate A because that's all it's going to bind. So by default, not only is it specific for the substrate, but for that reaction. Because remember, it's going to take that substrate and convert it to a product. So you can say an enzyme is specific for its substrate, but therefore it's also specific for that reaction. Okay, so good, it's specific. I saw recyclable, that's excellent. So remember, that means that the enzymes get used over and over and over again. They are not permanently altered. They are not consumed or used up in the reaction. That would be so inefficient, okay? So the enzyme here, if you can see my hand, the enzyme is gonna bind the substrate, it's gonna kick out that product, reset, bind another substrate, kick out a product, reset. So, so it just keeps going over and over. We're not constantly making the same enzymes over and over again. All right. Yep, you can tell that we're talking about an enzyme because they end in ASE. And then they are catalysts. Good. They are catalysts because they speed up reactions. Okay. So a catalyst increases the rate of a reaction. That's a fancy schmancy way of saying it speeds up a reaction. It increases the rate, and it does so by decreasing that activation energy. Remember, that's the amount of energy needed to actually start the reaction, to take A and convert it to B. Good. So last thing with enzymes that I really want to stress is we talked about how 3 things affect an enzyme: temperature, pH, and substrate concentration. So remember, these little square brackets in science, those mean concentration. So substrate concentration. So remember, y'all, when it gets really cold, the enzymes are not as active. Okay, why is that? How come enzymes aren't as active in the cold? Yes, because as the temperature gets colder, they just move really slow. Okay, so it's not that the cold denatures them. It's just that in the cold, the substrate and the enzyme are moving so slow that it takes a long time for them to find each other. Okay, now, as Ashley mentioned, in the heat, okay, the heat is going to make them go faster. So at this point, it's like if you shake a toy too much, right, it's going to break. So at high temperatures, then the enzymes are going to denature at some point, okay? So in the heat, enzymes can be more effective initially, but then if you kind of heat them too much, then they shake too fast and they'll break. Hold on, y'all. They locked my dog in here thinking she wanted to be with me, but she doesn't. Now she'll probably want in because I kicked her out. All right, so remember with pH, at low pH, it's an acidic environment, lots of protons, positively charged molecules. That's going to cause denaturation. At really basic temperatures, lots of hydroxide, negatively charged ions, that's going to cause denaturation, okay? Because remember, proteins are held together by disulfide bonds, by ionic bonds. So if you throw in a lot of extra ions to the mix, there's going to be a competition. And eventually, if there's too many positively charged ions in the solution, then the ionic bonds of the protein are going to get broken, okay? And that's going to cause the enzyme to fall apart. And then with substrate concentration, people have the misconception that if you just add more substrate, they're always going to work faster or more efficiently. And that's not true, because at some point, the enzymes get saturated, okay? So if you have 1,000 enzymes, as you increase the amount of substrate, yes, more of them are going to be able to work. But once you hit 1,000 substrates, well, now all of the enzymes are working. They can't work any faster. So if you add in 2,000 substrate or 3,000, the amount of activity is not going to change because there's still only 1,000 enzymes. So once you saturate the environment, if you look at the PowerPoint, the curve plateaus. Once we saturate our enzymes, activity plateaus, okay? So it doesn't just go up and up forever. All right, y'all. That's really kind of the take-home message with enzymes. Any questions about those? All right. So hold on, let me see what we did here, or what I did. I just put this together. Okay, since we covered a lot of microbial growth yesterday, this will kind of be a review for when we get to number 3. For number 2, I want us to look at our macro elements, okay, our macronutrients, same thing. Remember, macro meaning large amounts. Oh, hold on, I just saw Gardner's question. Yes, the third type of inhibition. Does anybody remember what that one is called? It's a good question. Let me erase some space real quick. Good, yeah, feedback inhibition. All right, so feedback inhibition comes into play when we have what's called a metabolic pathway. It's multiple reactions that happen sequentially. So maybe molecule 1 gets converted to 2, 2 will get converted to 3, 3 gets converted to 4. We'll stop there. Now let's say enzyme A is responsible for getting molecule 1 and converting it to 2. Remember, enzymes are specific. So that's all enzyme A can do, is take 1, convert it to 2. So that means we need a different enzyme to do 2 to 3. So I'm just going to call that enzyme B. But again, because of specificity, B can only start with 2 and make 3. That's it. So that means we need a third enzyme here that will take molecule 3 and convert it to molecule 4. So that would be a metabolic pathway. We have 3 reactions that happen sequentially, okay? So what this means is in order to make molecule 4, we have to have molecule 1, molecule 2, and molecule 3, right? Because that's what leads in to molecule 4. So normally when there's these type of pathways, the last molecule, molecule 4 in this example, is being used almost immediately, okay? So as molecule 4 is made, the cell uses it. But let's say the environment changes and the cell no longer needs number 4. Well, the cell has to stop making it, right? And so one way that that happens is as molecule 4 builds up in the cell, because it's no longer needed, it's not being used, what happens is molecule 4 can then act as an inhibitor to that very first enzyme. So the product is no longer needed in the cell. So the product eventually acts as an inhibitor to shut down that enzyme. And by shutting down the first enzyme, right, we'll no longer get molecule 2, which means we can no longer make 3, which means we can no longer make 4. So kind of the Chelsea definition is the product of the pathway, the product, shuts down the pathway that made it. Okay, so the product shuts down the pathway that made it by inhibiting that very first enzyme. Good, thank you for reminding me of that. Any questions about that? Okay, so for your macronutrients, I need you to remember why each of them are important, okay? So I want you to take just 30 seconds, because I know we're already at 5:20, take just about 30 seconds and try to remember, okay, well, which do proteins, do they have carbons in them? Yes or no? Do they have hydrogens? Yes or no? And put a check or an X or something if the element is needed to make that molecule. 30 seconds, ready, go. Yeah, you'll need to know. I think there's only one question about this stuff, but I'm gonna show you it's not near as scary as you think. So this is not what I would devote all your energy to. But it is something that it is important to understand why. All right, so I think that's been about 30 seconds. So let's go ahead and do this together. Let's see, I think I'm gonna use green for this one. I don't know why that sounds fun. All right, here we go. So y'all, I put carbon, hydrogen, and oxygen first because they are present in every single macromolecule. So you can just go ahead. Great. So those are easy, okay? And if you think back to your chemistry days, carbon and hydrogen pretty much always go together anyways, right? You can think of them like besties. There's tons of carbon-hydrogen bonds. All right, so with phosphorus, excuse me, phosphorus is a key component of phospholipids. Can y'all see that? Hold on a second, I can't see what screen am I sharing? I don't even know what y'all can see and what y'all can't see. Can y'all still see the Word document?
Yeah,
the Word, okay. Y'all can see that? Okay. Sorry. All right. Oh, this computer is going a little bit bonkers on me. Okay, sorry. All right, so we know phosphorus are in phospholipids, okay? And so the reason, I'm sorry. Come on, please work. Okay, there we go. So phospholipids are key to plasma membranes, right? And so we know that phosphorus is absolutely essential for all cells because we all have that phospholipid bilayer, okay? So phosphorus is also important for nucleic acids because nucleic acids have nucleotides, okay? And nucleotides were the sugar, the nitrogenous base, and the phosphate group. So we can go ahead, there we go. Now we're rolling. They work there as well. All right, so nitrogen is going to be important for proteins and our nucleic acids. Oh, and let me show you all this before I get too ahead of myself. I don't want y'all to be like, where did all of this come from? Let me see, where's, oh. Y'all, which screen am I sharing? There we go. So this is where it's coming from. Okay, so nitrogen needed for amino acids, that's for proteins. Nucleotides make up nucleic acids. And so that's, his setup is backwards for mine, so I am really on the struggle bus with this. My office will be set up soon. So that's why, like with phosphorus, we have it on phospholipids and nucleotides. So that's why we put them right there. Okay, and that's why we did the same for our nitrogen. Okay, so for sulfur, this is really only important for our proteins. So if you wanted to, and I guess had I mapped out this for y'all sooner, if you notice, proteins have everything but phosphorus. Nucleic acids have everything but sulfur. So you see kind of what I'm doing, and then lipids are missing 2 things, carbs are missing 3 things. So you could arrange this so that it's kind of more of a linear picture. But I would just pay attention, y'all. Give your brain a break instead of saying, oh, you know, proteins need carbon, hydrogen, oxygen, nitrogen, sulfur. Just remember this. Proteins don't need phosphorus. Nucleic acids don't need sulfur. Right? So then you only have to remember 1 thing instead of 5 or 6. Okay? So then we have 2 here and then 3 here. But this is why they are the macronutrients. And if we actually looked how much carbon is in the cell, there's going to be a lot more carbon than sulfur, right? Because sulfur is really only needed in bulk for proteins. Okay? So focus more on what they're not for, would be my advice. Okay. Now, as a kind of a quick review from last class or last review, go ahead and try and match these up together. Okay, now this part I did copy from a previous semester when I worried more about the exceptions to some rules. So go ahead and on G here, just cross out the grow a little. I was with science, it's always, you never want to use the term always or never, because right, there are always exceptions to every rule. And so when I wrote this, I think a year ago, I wrote this particular matching, I was much more concerned about wording things precisely, and it just confuses people. So take 30 seconds or so and do your best to match these up. They tend to be chunked together. So like the first, 4 are temperature. So A through D are also temperature. Oh, sorry, y'all. We also changed books. So this extreme thermophile is a hyperthermophile. They're the same thing, just some books prefer one term over the other. Make sure there are no other ones. That's okay. It's on there for a reason, though. So go ahead and just try and match it up. It's there for a reason. No, that's good. And I'll tell you why it's there in a second. Not to induce panic, I promise. All right, so for the sake of time, let's go ahead and start doing these together. So in the chat, go ahead and put what letter corresponds to a mesophile. Good. C. So remember, the human pathogens that we're going to focus on, most of them, like 99%, are going to be mesophiles. That's why they can infect us, because they grow best at our body temperature. We'll talk about listeria, which actually grows at cold temperatures, which is why we had the bluebell crisis. I swear this dog thinks she owns this house. All right, hyperthermophiles, which one are they? Like y'all already knew what I was going to ask. Perfect. So remember, thermophile means it loves the heat. Hyper pretty much means extreme, which is why some people call them extreme thermophiles. So it's just they love intense heat. So good. That's going to be about 95 degrees. So thermophile loves the heat, but not quite as hot. So which one's that going to be? Yep. And then our psychrophiles are psycho because they love it cold. Dog is killing me. Okay, good. So do we feel okay with the temperature ones? Okay. Really, in my opinion, the hardest part of this chapter is just all the terms. All right, so next up, the next, what, 5? Yeah, our oxygen requirement. So remember, when we talk about oxygen, we're not talking about this single oxygen that you find in these macromolecules. We're talking about O2, atmospheric oxygen. All right, so I haven't even looked at these. All right, which one is aerotolerant? No. Oh, aerotolerant. Crap. Yeah, I'm sorry. I'm sorry. In my mind, I was doing microaerophile, and I was like obsessed with E. And I'm like, why is nobody getting E? Sorry. Okay, aerotolerant. That means they can literally tolerate air. They cannot grow in it, but they're not going to die from it. Okay. So that means, yes, G. They cannot use it. They cannot metabolize with it, but it doesn't kill them. They can tolerate that air. And kudos for y'all, even though I was like, no, that's not it. Y'all are like, yeah, G. So good. Kudos to y'all. Okay, so now on to microaerophile, which I was obsessing on. Which one is that? There's the letter I was looking for. Yes. So remember, micro just means small amounts. So they still need air, right? Because aerophile, they love air. They need it. But the micro means they need it in less than atmospheric amounts. Good. So less than normal amounts of oxygen, but it is required. All right, obligate aerobe. Which one is that? Good. They need it. They must have it. All right, obligate anaerobe. Which one is that? Good. So remember, this is because they do not have catalase or peroxidase or superoxide dismutase. So they can't handle any kind of toxic forms of oxygen because they don't have an enzyme to break it down. So then by default, our facultative anaerobe is 8. Remember, these are our switchers. They're able to grow, which means to divide in either situation, with or without oxygen. Excellent. All right, so now go ahead and let's do the last 4. So alkalinophile, which one is that? Good. So for y'all, to remember, let me get my speech together. What y'all have to remember is that high pHs are bases. We also say that that's an alkaline environment, hence the alkalinophile, because it's an alkaline loving organism. So a basic pH. Now capnophile, which one did y'all put for this? Good. Okay. Now, let me tell you this. We did not talk about capnophiles this year, but here's one. So it's not going to be on your test, but here's why I kept it on this sheet. When you, oh, wait, that's very true. That's exactly where it comes from. When you're taking the exam, okay, now I'm not going to put anything on there that you've never heard of before. And believe me, people will email me and be like, what was that? And I'll fix it. If I ever do that by mistake, I always go back and give everybody credit. You're not going to be punished for my mistakes. But you'll get there and you'll be like, oh, have I seen that? Oh, I don't know. But the reason why you shouldn't panic because there's process of elimination. And because you do know what an alkalinophile is, an acidophile, a halophile, by default, right, you can infer what a capnophile is. If you're ever on there on the exam or whatever, and you're just like, oh, I don't remember, I'm not sure, well, rule out answers that you are confident in, okay? If you're anything like me, like I have anxiety, and I was the kind where I was like, what was, I don't remember, oh my gosh, I don't know it. In my mind, it was just chaos. But then it's like, take a deep breath. What do you know? And what can you rule out? Okay, so this was here just to show you that even if you're not familiar with something, it doesn't mean you can't answer it. Okay, but don't worry, capnophiles are not actually on your exam. Okay, so acidophiles, which one is that? Good, low pHs, below 6. Ooh, oh, I forgot I left in the, yeah, hold on, Ashley, I'm gonna answer that. Give me 1 second. Let me just finish filling this in. Then halophiles, those are gonna be our salt-loving organisms. So capnophiles tend to also be microaerophiles, because if you think about it, maybe in the lecture notes, it still talks about the candle jar. So like if you think about like a candle, you put it in a jar, right? If there's lots of oxygen, what happens? But then think about what happens if you put the lid on it, right? Doesn't the flame normally, excuse me, go out? And because it's like it needs the, excuse me, the oxygen. So microaerophiles need small amounts of oxygen, and by default, they normally prefer large amounts of carbon dioxide, because a lot of times those go hand in hand, like they're inversely related. So if something needs a little bit of oxygen, it probably needs more than normal carbon dioxide, and vice versa. So something that's classified as a microaerophile is typically also classified as a capnophile. Good memory, Ashley. Okay. I thought of something I wanted to tell you. Oh, let's do a math problem, because you'll probably see that again. So I'm going to wing this, y'all. So I'm going to try and make the math nice. Let's say you're in the lab and you have a starting culture of 200 cells. You have a generation time of 10 minutes. How many cells will you have in 40 minutes? Now don't type it in the chat, but don't push enter yet. I don't want one person's answer to sway anybody else's. So put your answer in the chat, but don't push enter yet. Okay, so put your answer in the chat. Don't push enter. And I'm going to count us down. I'm going to say 1, 2, 3, go. And on go, you're going to push enter. Okay? So get your number in there. All right. 1, 2, 3, go. Yay! All right, let's work it out together. So remember, whatever you're starting with, we call that T0. Because at T0, that means before you start the timer. So what you're starting with. So we're going to have 200 cells to begin with. So in 10 minutes, that's our first generation. So we call that T1. So after our first generation, our population doubles each time, right? So this is at 10 minutes. And so our population is going to double to 400. At 20 minutes, that's our second generation. So that's why it's T2, second generation. We're now going to have 800 cells, because again, it doubles every generation. 30 minutes would be our third generation. So our 800 cells will double to 1600. And then at 40 minutes, that's our fourth generation. So now our 1600 cells each divide. So that's going to give us our 3200 cells. How do we feel about this? Are we okay? Remember, your 200 is what you're starting with. Okay, don't count that as T1, is what you're starting with. Yep, exactly, Amanda. All right, so real quick, go ahead, sketch a bacterial growth curve and label the 4 phases. I'm gonna start. Okay, just because I'm short on space, I didn't write the name. I kind of numbered them. All right, y'all. So what is the first phase? When you take cells on your loop, you stick it into maybe a new broth or whatever, put it in the incubator. Good. First is going to be our lag phase. And it's called that because there's a lag in growth. Okay. Notice on my picture, it's a straight line. That means the number of cells is not increasing or decreasing. Right now, they are just adjusting to their new environment. Okay. Once they get comfortable, they're like, oh, hey, plenty of space, plenty of nutrients. I can start doing my thing. They're going to start undergoing binary fission. Okay, so remember, with binary fission, you have your cell. It's going to elongate. As it's elongating, the septum, where it's kind of bending inwards, the septum starts to form. The DNA is going to be replicated, okay? And then as it continues to pinch inward, as that septum continues to form, one copy of the DNA goes to the left side, the other copy to the right, and eventually they pinch off into 2 cells, okay? So that's the division. This is what we mean when we say growing, bacterial growth. We're not worried about the cell actually getting larger. They don't grow, like humans do. When we say growing, we mean dividing. The population is growing, okay? So once they start dividing, they do so exponentially, which is also called logarithmically. So the second phase, exactly, Shelby, is our log phase because the population is growing logarithmically. And then go and put what number 3 is called. My computer battery is bad. It's about to die. It was fully charged 45 minutes ago. All right. Yes. The third one. Oh, he's not on the outlet. Hold on. I'm going to talk. So if you can't hear me, though, let me know. The third stage is stationary. Okay. Notice that, again, there's no bacterial growth. There's no net increase or decrease in the number of cells. And that's because this is the point where the cells are running out of room. They're running out of nutrients. And so there's a way for bacteria to detect that it's too crowded. And so they stop dividing. So they're kind of in survival mode here. They're just hanging out, chilling, hoping for a better environment. Hold on, I'm going to play this thing. Okie dokie. So stationary because growth is stationary, okay? But like any living thing, right, it can't just wait forever, okay? It needs food. In this case, they need to divide. And so eventually they enter death phase. And at this point, they're dying logarithmically as well. All right, any questions about this, y'all? How are we feeling about Unit 2 at the moment? We've gone over growth pretty in depth Are there any questions about that section? Good. Yeah, this one's, it's not quite as dense as Unit 1, in my opinion. There's still a lot. There's always going to be a lot for micro, but at least this stuff tends to be a little bit more, it's just not so abstract. Okay, so originally with this flowchart, I was going to have it all be blank, but like I said, I only had about 20 minutes to throw this thing together. So I filled in more of it than I had wanted to, but this will allow us to talk about it more than just spend a bunch of time trying to build it. But what I do want to show y'all real quick, because being able to create this yourself is going to be very helpful. So what I wanted to find is to show you how I did this briefly, very briefly, because I know we are running out of time. All right, so I know it's tiny. Here we go. So with this, right, controlling microbial growth, we talk about 3 ways. Physical means, chemical means, and antimicrobials, okay? So that's why, and as you can see, I didn't draw all the lines because I didn't have time. With controlling microbial growth, these are the 3 aspects we're talking about. So you might want to go ahead and put in the lines. Clearly, I draw very good straight lines. Shaky hands, I could never have been a surgeon. So when it comes to the antimicrobials, you could build this whole thing out, okay? But we did a mind map on Friday, so I'm not going to do that, okay? But when it comes to physical methods, if you look on this PowerPoint slide, I tell you we break it into 3 sections. We spent most of our time for temperature with heat, but think about what you do with your leftovers. You put them in the fridge, right? Because that's colder than bacteria need. So if you want to, you could take this and change it to temperature. Now I didn't make this on landscape orientation, so I was kind of limited on space, but you could easily make that temperature, have it branch off into hot and cold if you wanted to. It's completely up to you. Because our conversation focused mainly on heat, that's just what I did for this one, okay? So for physical methods, again, we're talking about the physical environment. Oops, whatever, I'll just get a purple. I just wanted that to go away. And so with the physical environment, here's the 3 that we talked about, okay? And again, it's just on this PowerPoint slide. And then say, okay, well, with respect to heat, we talked about moist heat versus dry heat. And from here, you could say, okay, moist heat, boiling, right? We talked about pasteurization. So you can build it out that way. Same with dry heat, ovens, Bunsen burner, okay? So again, I was just trying to get this started for you and kind of a jumping off point to talk about. Jordan, did you want to mention your question?
Yeah, so wait, I might be getting these confused, but on the last review session, we did, for physical, we did temperature, tonicity, oxygen, and pH. And then I was curious if we were going to add filtration and radiation, but I see you added them on this one, but then the other ones are listed as well.
Right, okay, so hold on, my sound is low. So what we did yesterday was focusing on what microbes need to grow. So they need, do they need a lot of salt or no? Do they need a certain pH or no? Here we're talking about how do we prevent them from growing? How do we prevent our media from being contaminated? So this is more controlling their growth rather than what they need to grow. Does that make sense?
Yeah, that makes perfect sense. Thank you.
Okay, now, but I do want to kind of build off what you're saying because this is limited. I could have like a whole class, like a semester on controlling microbial growth because we do use pH to control their growth, right? If I told you that E. coli is a neutrophile, so it needs a neutral pH, well then if I go and I take my food and I squirt it with lemon juice, I've now created an acidic environment. So I've killed any E. coli on that food. This is one reason they sometimes, like with seafood, they'll give you lemons, citrusy things, and you squirt it on there. It can help with taste, but it's also ensuring that any little microbe that might be on there is going to die. So this is a limited conversation on control, but that's the big difference on your question for control versus growth. All right, so I want to just point out a couple of things from here because I know we're running out of time. So when it comes to heat as a physical method of control, moist heat versus dry heat. Remember, y'all, because moist heat involves water, water naturally starts breaking things down. So you don't need as high of temperatures. You don't need as much time when you're using moist heat because that water and the temperature are working together. When you use dry heat, you need higher temperatures and longer times because it just takes longer for things to break down when heat is the only thing happening, okay? So remember with pasteurization, that was important for like beverages that we drink because you can't expose them to heat for too long or else you're gonna mess up the temperature, the flavor of the beverage. So we talked about the two types, high temperature, short time versus ultra high temperature. So remember, this is why some like milk or creamer, if you like put cream in your coffee, some of it's in the refrigerated section, some sitting out on a shelf at room temperature, and it's perfectly fine. But it's because they were disinfected differently. Things that sit on the shelf were blasted with really high heat to reduce the number of microbes present. So there's less microbes in the room temperature cream compared to the one that needs to be refrigerated. That's why you refrigerate it, to prevent the existing bacteria from growing, because there's just more in there, okay? So make sure you pay attention to the two types of radiation, ionizing versus non-ionizing. Ionizing physically breaks the DNA, whereas non-ionizing causes those thymine dimers, which, oops, that's a D, which warps the DNA. It alters its structure, so DNA replication can't happen. So they're both targeting DNA, but one physically destroys it, and the non-ionizing is like the passive-aggressive form. It's going to cause the DNA structure to warp, so DNA replication can't happen, okay? So a little bit different outcomes there. For the chemical methods, remember we mentioned alcohol. We talked about two different alcohols. Their big issue though is they evaporate so fast. So that's good if you're putting it on like a flesh wound because it won't sting too long. But if you're trying to clean a really dirty table, most of it's going to evaporate before it does anything. Remember, hydrogen peroxide is really good for fomites, inanimate objects, because a lot of bacteria at the surface of your skin have the enzyme catalase, which breaks down peroxide. So if you try to just dab a little peroxide on a surface flesh wound, those bubbles are really the bacteria laughing at you because it's them breaking down the peroxide, not them dying from it, okay? And then remember, surfactants, these are amphipathic. Oops. Amphipathic, and that's why a lot of soaps and detergents work so well. Because remember, amphipathic means the molecule is hydrophobic, so it's going to stick to all the dirt, the oils, things like that. Hydrophilic means it's going to pull it off your skin and down the drain when you add water. Okay, so I know this was a super quick conversation on those. Those are kind of like the highlights, but make sure you go back and check out the types of pasteurization. Remember autoclaving. That's real important for lab. So what I love about this unit is I can start giving you more like scenarios and more real world applications, right? When the water companies, when the water's not being cleaned, they tell you to boil their water. That's why. Okay, so real quick, y'all. I know I only have 3 minutes. Please log off when you need to. We are going to walk through this, but because we did talk about it a little bit yesterday, we're going to go a little bit faster, but there were a few things I kind of glossed over yesterday. So please, if you need to go at any point, I'm not offended. All right, here we go. So I recommend kind of filling this out on your own from your memory, what you can remember. So remember, our first step of aerobic respiration is going to be glycolysis. And I would write smaller. I made these bigger so you can take notes in them. but I can't because I write too big. Ah, poop. Hold on. How is this? Is that better? Okay, at least this happened at the end. Okay. So I'm going to then just talk, and I'm going to suggest y'all kind of jot down what I'm saying. So remember, step 1 is going to be glycolysis, but there's a few things you want to remember about glycolysis. So I would write this part kind of small, or you can put it in the notes if you want. So glycolysis is the step of respiration that happens with or without oxygen. I'm going to make a note of this. I'm going to not move the screen. That's supposed to be a with or without oxygen. That's very unique because with aerobic respiration, you need oxygen. Is this working? Can you hear me?
Yeah, I can hear you, but you were kind of going back and forth.
Yeah, I think it's because I'm trying to write at the same time. So I'm going to put my pen down and we're going to try this again. All right, so glycolysis. Remember, this is the step that happens with or without oxygen. This is also the step that happens in the cytoplasm of all cells. Okay, it's the cytoplasm. All right, so step 2 is going to be the linking step. And so remember, now the point of this is to link glycolysis to the Krebs cycle. The third step is the Krebs cycle. And this is happening, I guess for both step 2 and 3 happen in the same place. The cytoplasm for prokaryotes, because that's all they've really got. And it's happening in the mitochondrial matrix. So we're now in the matrix or the mitochondria for our eukaryotes. And remember, the matrix is just the middle. It's the middle of the mitochondria. And remember, as of now, as of step 2, oxygen is absolutely required. That's why it's called aerobic. No, step 2, 3, and 4, actually. That's why the step 1 was unique, because step 1 is the only one that happens in the cytoplasm for our eukaryotes. The rest happens in the mitochondria for the eukaryotes. For the prokaryotes, steps 1 through 3 happen in the cytoplasm. Step 2 and 3 is the matrix. So it's the mitochondrial matrix. So hold on, let me sketch this. So this is our mitochondria. That's the outer membrane. That's the inner membrane. This middle part, that's kind of like inside the little wall here, that's the matrix. So step 2 and 3 happen in this matrix, this center part of the mitochondria for eukaryotes, because remember, prokaryotes don't have organelles. All right. So step 3, so both the linking step and Krebs cycle happen in the matrix. All right, step 4, oxidative phosphorylation. This happens in a membrane, okay? Step 4 requires a membrane. So for our eukaryotes, it's in this inner mitochondrial membrane. The highlighter is really big, but it's just those folds. So for our eukaryotes, it's in the inner mitochondrial membrane. For our prokaryotes, the only membrane they have is the plasma membrane. So remember, y'all, for this last step, this is where the electron transport chain is and where ATP synthase is. So that's why for eukaryotes, that's why this inner membrane is all folded. They're trying to squish as much membrane inside as possible, because the more inner membrane they have, the more electron transport chains, the more ATP synthases they can have. So essentially, the more inner membrane, the more energy the cell can produce. I'm sorry, y'all, I forgot to turn the video off, so I'm like up close and personal when I'm drawing on here. Sorry about that. Okay, So what you want to really remember for this, down in the notes section, what I would put, oxygen is your final electron acceptor. And then I would make some sort of note about the location. Most of this is in the mitochondria for the eukaryotes. Most is in the cytoplasm for the prokaryotes. Okay, now we mentioned the exceptions to that, but for the most part, mitochondria for eukaryotes, cytoplasm for prokaryotes. And definitely remember, oxygen is the final electron acceptor. That's why this is an aerobic process, because oxygen is required from step 2 until the very end. I'm trying not to move this too much, so my handwriting is even worse. I swear this little weenie dog wants to be a snack. It is tempting Kaya. Okay, so with anaerobic respiration, what people typically misunderstand is they think it is so different from aerobic respiration. But do we agree, y'all? Respiration is respiration. They can't be that different if they have the same name, right? What's the big difference? The anaerobic part, okay? So I'm gonna fill this in real quick and then I'll talk about it. It'll only take a second. Okay, so notice right off the bat, steps 1 through 4 are the same. What I tried to do was put a squiggle there for steps 2 through 4. The linking step, the Krebs cycle, oxidative phosphorylation, they're a little bit different in our anaerobic organisms. They're still there, but they're not as efficient. So we say it's an abbreviated Krebs cycle. It's shortened. It's an abbreviated linking step. So it still has them. They're just quicker, smaller, and they don't give you as much energy. Okay, but they still exist. So the big difference between these, aerobic uses oxygen as the final electron acceptor. Anaerobic respiration does not. It uses like sulfate or nitrate, something else that loves electrons, but it's not oxygen. Okay, so these are very similar processes. Okay, now fermentation is where things get different. So fermentation has glycolysis. That's the one thing that all of these processes have in common. They all have glycolysis. So fermentation produces the least amount of ATP because it only has glycolysis, which only makes 2 ATP, okay? You don't need to memorize these numbers, but just to put it in perspective, from 1 molecule of glucose, aerobic respiration makes upwards of 32 ATPs. Anaerobic respiration is about half, it's about 15. Fermentation is 2. Okay, so fermentation is not very energy efficient, but it's all some organisms can do. So remember, fermentation is an anaerobic process. So that's why if something can use oxygen, it's able to do respiration, it's able to get a lot more energy. And actually, you should have put anaerobic down here in like the notes section. So I would note that it's an anaerobic process, and I would note that it doesn't make a lot of ATP. And anaerobic makes about half of aerobic. All right, I'm sorry to have kept y'all over. Do y'all have any questions that I can answer before we say goodbye?
Not a question. I'm super thankful for these reviews, though. Seriously, thank you.
Oh, you're absolutely welcome. I really enjoy them. I think they're really, really fun. Because I get to do more, like the hard part, and y'all don't have to listen to me babble on here if you don't want to. Like in class, people, if I don't use the PowerPoint, people tend to get more confused, and that's understandable. You're hearing it for the first time. So using flowcharts and things isn't always as effective for introducing stuff, right? And again, with the PowerPoint, it helps people keep their notes straight. But when we do reviews, and in theory, you've studied this stuff, doing things more like this, which I think are more interactive, more fun, more visual, it tends to help people connect the dots once they understand the dots. So I love flowcharts too. I'm so glad not every group of students is as responsive to it. So I'm glad y'all are flowchart people. All right, y'all have a fabulous evening. I know y'all are gonna do awesome on this test. If you have any questions, I've got the Remind app. Remember, this weekend is not good for email. So yeah, Jordan, if you want to stick around, we can look over your quiz. Absolutely. Okay, I'm gonna quit sharing my screen so that I don't show people everybody's stuff. Y'all have a fabulous weekend. This mouse is not cooperating. Where are you? Yeah, if you can send it Remind, that's fine. And if you need to, you can use, you can like do a voice recording if it's easier than typing out your question. That is an option too. All right, talk to you soon, Taylor. All right, looks like it's me and you, Jordan. So let me get your, is this the unit quiz?
Yes, ma'am.
All right, give me 1 second to get all my windows straight. I have all my like screens go from left to right, and my husband's a weirdo, and his goes from right to left, and it's really
It's
not.
probably throwing you off.
Yes, and maybe how his computer is set up, maybe it does go left to right, but how mine is connecting, it's not working in my favor.
You're doing better than I
Oh,
was. I'm not technologically savvy at all in the slightest.
Oh my gosh, it's been a struggle. I had to kind of figure it out when COVID happened and we all went virtual, but thankfully my husband's in IT sales, and so he at least knows some computer stuff. He's got a crazy cool setup. I wish I knew how to do it. Okay, let me see. Come on, computer. Oh, did I stop the recording? Oh, poop. Hold on, let me go get that first. Sometimes I just exit and I don't stop the recording, which means everybody can still hear us. Let me make sure. It says you're already in this session, but I'm not in there anymore. Amy, are we still in there? Holy moly, we are.
I think so. It looks like it's still recording.
Oh my goodness. Hot Mess Express.
No, you're good.